Okay. Is everyone here? And okay. So let's see. So we have about an hour and a half. Wait, no, three. Tama. Hour and a half before your next class. So let's try to. I'll try to get to cover as much as I can. This is already a bit shorter from the five videos that you should have been able to see in Canvas. So and I'll just go through the main parts of those videos and to stress certain points. But maybe rely on those recordings for more details. And if you have any questions from those videos, this is the time that you could ask them as well. Also, in the Canvas as well for the in the discussion boards, you can ask me there so that I can respond. So let's see. I'll share my screen. Oops. Let's see. Wait. Okay. Okay. Can you see the slide?
Yes po, Doc.
Yes.
Okay. Thank you. So let's see. Let's go ahead. So the outline for this lecture would be to go. This is an introduction to neuroanatomy, and it's like a set of five videos that you had in Canvas. First is the nervous system organization in general. Then the three lectures on the central nervous system, the brain, the brainstem, and the spinal cord, and the part of neuroanatomical localization. Um, the last one is important for the course actually to have a perspective on all the lectures that you will have in neuroanatomy subsequently. Let's see. I'll just minimize my. I'll let's see. I'll stop my video para I won't have any problem with um, lagging. Anyway, so these are the outcomes, learning outcomes that are posted in Canvas. So first is to describe the organization of the nervous system, defining the common neuroanatomical terms, naming the principal divisions, which are the central nervous system and peripheral nervous system, and their component parts. So that's for the first lecture. Then the next would be the divisions of the central nervous system. That is the brain and spinal cord. And the brain being divided into forebrain, midbrain, and hindbrain, and also a description of the general topology of the cerebrum into lobes, gyri, and fissures and sulci, as well as mentioning the principles of the functional cortical areas and the Brodmann areas. And then the brain stem, which is part of the brain, identifying its parts, what are the external landmarks, and as well as the internal features of meaning the cross-sectional anatomy of that brainstem at different levels, with the three parts, the big brain, pons, and medulla oblongata. And then the spinal cord, the gross features, the developmental changes with respect to its relationship to adjacent structures, that is the spinal column, and then the cross-sectional anatomy of the spinal cord, division in gray and white matter, the differences between specific cord segments, that of the cervical, thoracic, lumbar, sacral, uh, the structures that can be identified like the fasciculi, funiculi, and fiber tracts, and then to end with the rexed laminae. The video also talks about the principles of neuroanatomical localization. I will not cover this anymore today. Just watch the video itself and to get a perspective on those, as I said, on the coming lectures, so that you could the idea of our neuroanatomical course is towards identification of possible site affected in cases of patient with neurological deficit. No, so this is what a neurologist would do first in identifying when a patient comes to them, and this we at your level you are not expected uh, to diagnose the cause yet, but you just have to identify where in the nervous system a particular if a patient manifests with neurological deficits, where would be the most likely location of that injury. So anyway, so those are the some principles that I put in that video, and it will not be comprehensive, of course. So it will take time, and it will do that more in the second year in the bridging of the neurology one. Okay, these are the references that I had for this lecture and the video. So Snell's Clinical Neuroanatomy, Haynes Neuroanatomy in Clinical Context is a useful atlas, especially for the lab when looking at segments of the brain, sections of the transverse and coronal sections of the brain, as well as the spinal cord and the brainstem. No? So very useful for us in locating particular structures. Also useful is the Netter's Atlas of Neuroscience. The images in the regular Atlas of Netter had been is limited, no, the plates there. So they actually what they actually in the older edition of Netter, they actually removed a number of them and put them together in this separate atlas of neuroscience. No? So most of the uh, images that pertain to neuroanatomy are in this particular Netter's atlas, no, for neuroscience, and we're good to get that one if you don't have the copy of Haynes. There's an old book that may still be useful for you, which is the Manter and Guts. This had stopped. The last edition available is in the 2003. They haven't updated this, and because the publishers don't want to update it. There are some parts of it that can still be useful. And in some of our lectures in the neuroanatomy, some they have this particular book has a particular help there in understanding certain concepts that may not be immediately understandable in Snell or in Netters. This last reference, localization in clinical neurology, that was the main reference I had for the topic on neuroanatomical localization. So that's a whole book about localization. So you don't need to have to get one, but maybe borrow from the library and to be able to read that particular chapters, the first few chapters that pertain to the principles of neuroanatomical localization. Okay, so with that being said, let's go to the main bulk of this today's lecture. So the nervous system is divided into two major divisions, the central nervous system and the peripheral nervous system. On one hand, the central nervous system is, wait, okay. Central nervous system is divided into the brain and spinal cord. So just that. And then everything else that is not brain and spinal cord is peripheral nervous system. These are the cranial nerves which come off from the parts of the brain, and then the spinal nerves that come from the spinal cord, and then the associated ganglia. These are nerve cell bodies accumulated outs that are where nerve cell bodies of the neurons are in the peripheral nervous system outside of the central nervous system. Okay. Now, let's review the parts of the typical neuron. So you have the uh, nerve cell body or the soma, which is, teka, and try to see if I can, no, I don't have a pointer. So anyway, so the nerve cell body or soma is where the nucleus is, okay, of the neuron. It has dendrites. These are nerve processes that carry impulses towards the nerve cell body. Just the small ones there in this image. And then the axons are the nerve cell processes that carry the impulses away from the nerve cell body. Okay, the nerves, these axons may either be myelinated or unmyelinated, whether there is developed myelin sheath around them. Now, let's some review some of the definitions of common anatomical terms and concepts that we would use for the neuroanatomy course. So are the concepts of ganglion and nucleus, or ganglia plural or nuclei. Okay, now this would depend on the location of nerve cell bodies. So these are both nerve cell bodies. Okay, collection of nerve cell bodies. When they are collected outside of the central nervous system, meaning in the peripheral nervous system, they are called ganglia. Okay, so which you would have seen in the gross lab, like when we were in the head and neck, you have seen um, the trigeminal ganglion in the near the jaw, or the in the spinal column you have the sympathetic ganglia, no spinal chain, no sorry, the sympathetic trunk, no. So and then of course the dorsal root ganglion. So you would have seen that as well. So those are ganglia. These are outside of the brain and spinal cord. Now, there are, if these nerve cell bodies instead are located within the central nervous system, meaning they are collected within the brain and the spinal cord, they are called nuclei. So now, that is depending on the location of the nerve cell body. Now, remember that like by convention, when we're saying in the course, say if we ask during the course that where is the neuron, okay, what we actually mean is the location of the nerve cell body. Okay, so even though technically the neuron is the whole extent of its parts from the tip of a dendrite down to the tip of the axon, so that's the whole neuron. But when we ask regarding location of the neuron, what we actually refer to is the location of the nerve cell body. Okay, just remember that. Um, okay, next. Now, one possible confusing terminology in particular is the case of the basal ganglia or basal nuclei. So this is a collection of nerve cell bodies. Wait, okay, should share. Okay, ayun. So the um, if you look at the image on the right, there's this triangular pizza-like thing with three uh, collections there. So those are the basal nuclei or basal ganglia, no? So on either side. So those are they are located in the brain, deep in the brain. So this is a coronal section of the brain, no? So you see, so those are two like pizza wedges on either side. So those are the basal ganglia or nuclei. So therefore, they are in the central nervous system. So in that case, what should be the more proper term to use? Basal ganglia or basal nuclei? Ano? Anyone?
Basal nuclei po.
Yeah, yeah. So basal nuclei. So the newer um the newer textbooks already use the basal nuclei, no? Yeah. And we will be most often use that term preferably during the course. So, but if by mistake or somehow, or if you still see basal ganglia, then just know that they're referring to the same structure. Just in case you see it in the board exam as well, that's the same thing. No? So, because the examiners may use the older terminology. So, let's see. Wait. Okay, so poll question, maybe not apply the poll, let's just answer it acclamation. So the trigeminal Gasserian ganglion is part of the blank nervous system. Ayan, pinol na pala. Ayan, sige, sagutin nyo na nga lang. Okay. So most of you answered central. Peripheral pala. Peripheral, 70%. Okay. So the answer here is, oops, peripheral, no? Kasi remember the term, it's ganglion. So remember, ganglion in the peripheral nervous system, nuclei in the central nervous system. Yan, okay. Ang naging exception lang yung sa basal ganglia problem na yan. So, but in principle, basal nuclei na nga talaga yan. No? Okay? So, even for that, a clue, even if you don't remember what the trigeminal ganglion was, but if you sort of like can imagine it, it's outside of the brain, it's outside the skull. No? It's before it divides into the three nerve branches of the trigeminal nerve, of the ophthalmic, maxillary, and mandibular branches. So that's already outside of the skull. So that means it's outside of the brain. So ganglion, peripheral nervous system. Okay, next. So in the areas of the central nervous system can also be differentiated according to the appearance on fresh unstained specimens into white and gray matter. So white matter in this picture, so you see them in so this in coronal section of the cerebral hemisphere. So the white matter is the one in the middle, the white clear area, and then the darker part in the periphery. That's the gray matter. You know? So the white matter is made up of nerve fibers primarily. So, and what makes it appear white is because of the myelin sheaths that surround the myelinated axons in these areas. So since there are mostly nerve fibers, so and myelinated fibers, axonal fibers at that, so they appear white. No, so then the gray matter do not are mainly composed of nerve cell bodies. And remember, nerve cell bodies are not covered by myelin. Okay, so they don't have that sheath, so they will appear gray, no, in this picture. So you so just compare the arrangement of the white and gray matter in the brain on the topmost section. And it's hard to see appreciated in the brainstem cut, but look at the one of the spinal cord below. So there is a switch. So it appears to be a switch in which the gray matter now is inside, and then the white matter is outside on the outer surface. No, uh, that is the how it appears in the spinal cord. You will find out why that is in the lecture on development of the central nervous system, no? From the neural tube. So so how there would be gray matter outside on the outside part with regard to the brain. Now, now on section, and there are some of the sections that you would see in the lab, also pictures in your textbook of stained specimens of the brain and spinal cord. If they are stained with for myelin, the image would reverse. So this is one image there of the spinal cord section. So white matter will appear dark, and the gray matter would be lighter. So as in this picture, so since the stain is picked up by the myelin, so it looks black. No, so the white matter is black, gray matter is white. You know. So you just have you have to after being familiar with what how sections look like, you know that already that this is a spinal cord section, and you know that this is a stained specimen because white matter, which is outside on the periphery, should not here appears black. So that means it's a stained specimen. No? Okay. This is can also be done for the cross sections of the brain as well. So, but in this example, you see it in black. Okay. Uh, So let's go to some terms on how white matter is organized. Let's just go quickly through these terms. No? First is corona radiata. The corona radiata is a sheet of white matter formed by fibers projecting like a fan to and from the cerebral cortex. So if you look at this image on the cerebral hemisphere on top, I'll just animate here. So it's like a little fan there. So there's actually fibers that from the cortex down towards the deep into the cerebrum, and as well as the deep in the cerebrum going to the cortex because of these are the fibers um, that communicate with the cerebral cortex, and at that particular level it's like a sheath, no, it's like a fan, so it's like a corona radiata. It's different from the corona radiata that we have from the lecture of Dr. Rivera earlier, that surrounds your ovarian follicle. Okay, that's a different corona radiata, no? But the same term, but different structure here. Okay, uh, so you may appreciate this better on when you do the lab already for you. Now, peduncle is a stem-like connection. So peduncle means stem, no? Um, and then this is between two areas of the cerebral, as of the central nervous system. So an example with is this is the cerebral peduncle. Let's see here. Ano ba yun? Teka, hindi, I can, ano, alam ko na. I will, oops. Hmm, ay, okay. Yan, okay. So this is the, ano, oops, I will go back. Wait, yeah, okay. Okay. So this is the cerebral peduncle. Yeah. So the cerebral peduncle connects the cerebrum to the brainstem. So this is the brainstem, and then cerebellar peduncles are connections of the brainstem to the cerebellum. So the cerebellum is not in this image. This is primarily the brainstem. So these are the peduncles. This is the superior, middle, and this is the inferior. Sorry. This is the superior. Sorry, middle, and then and inferior. These are cut in the same part, but this is actually the inferior. Okay. Uh, let's see. I will remove my. Okay. Next. Oops. So So the brachium is an arm-like connection. So it's similar to the stem of the peduncle. This you may use these particular terms for particular structure. So the brachium conjunctivum is also another term for the superior cerebellar peduncle. And the brachium pontis is another term for the middle cerebellar peduncle. So there are they can you can use certain terms for you can encounter certain particular um, terms for the same structure. No? But we'll just get the idea that of the derivation of these terms. Now, lemniscus refers to a band of nerve fibers, like a ribbon. Yeah, that's what lemniscus means. So this used to refer particular to some parts of the sensory tracts within the brainstem. So in this image on the right, this is like the connection of to the cerebrum, and then from the spinal cord, there are fiber tracts, no, yung in different colors here rendered in there's purple, blue, and green, no, and there's a little and little red here on the side as well. So they when as they go up when they reach the brainstem, which is this part where the in the middle part, uh, so they form the lemnis. These tracts form lemnisci, that of the medial, spinal, and lateral lemnisci. So and they because they appear like they go together like a band, so that's why it's called a lemniscus on each one. So they go up the brainstem until they reach the area of the thalamus here, where is in the right smack in the middle of the cerebral hemisphere. Okay. So yun. And this cross section image on the left. So this is a cut on the level of the pons. So that's how these lemnisci are arranged. So they're in a particular position. And you will be able to identify them perhaps later in when you go to the cross sections of the brainstem. So that's lemniscus. So they are made up of white axons of ascending tracts or sensory tracts. Now, fasciculus and funiculi, these are terms that are used for parts of the spinal cord. Okay. So, might as well, since we're here, let's look at the organization of the spinal cord already. No, so the spinal cord is the white matter is arranged into pairs of funiculi, meaning cord-like bundles of their fibers, where you have uh, where some of the fiber tracts are located in the spinal cord. So you have the dorsal funiculus, so or posterior funiculus. You have the lateral funiculus or funiculi on either side, and then the ventral or anterior funiculi. So right and left. Now, and they are divided or separated by particular grooves or sulci or fissures. So this first sulcus is the dorsolateral or posterolateral sulcus, which separates the dorsal funiculi from the lateral funiculi. So that's also where the uh, dorsal roots come into the spinal cord. So that's sulcus. No? Now, the ventrolateral or anterolateral sulcus, which is here in magenta, that's the one plane. You have the dorsal or posterior median sulcus that separates the dorsal funiculi on either side, right and left. And then you have the anterior median fissure that separates the two ventral funiculi. So this is a deep groove. That's why it's called the fissure rather than sulcus. No. So the in with referring to this structure, these grooves, we use the fissure for this particular structure, ventral or anterior median fissure. And the other ones we use sulci or sulcus singular. Okay. Now, at certain spinal cord levels, there's another sulcus that appears, especially in the cervical and upper six thoracic spinal cord levels, and it divides the dorsal funiculi further into two bundles, which is the posterior intermediate sulcus. Okay, or dorsal intermediate sulcus. So what are these? How does it divide the dorsal funiculi? So you have a medially located fasciculus gracilis or gracile fascicle, and the laterally located fasciculus cuneatus or cuneate fascicle. Okay. So, yeah. So the term fascicle basically refers to a nerve bundle, no, in the central nervous system. And it is used also in other nerve bundles in the brainstem and spinal cord. Like there is a medial longitudinal fasciculus. So you will see that later when you're studying in the neuroanatomy. And also here, there is a posterolateral fasciculus of Lissauer or Lissauer's tract. If you look at the image pointing to the violet arrow on the posterolateral sulcus, there's this dark band there line. So that's where the uh, just lateral to that dark line, that's white there. Yun, that's the posterolateral fasciculus of Lissauer. No? So it will have a role in the general somatic afferents or sensation from the body, from the passing through the spinal cord. We'll just have that in another lecture. So remember, this image is a cross section of the spinal cord. Okay. And thus the funiculi and fasciculi are also cut, transected in cross section. So you don't appreciate the cord-like or bundle-like feature. So just imagine these curved bundles here. So this is another image from Netter. So that's in a little bit more far. So you see them as a column going up along or down along the course of the spinal cord. They appear like cables, no, or columns on along the spinal course of the spinal cord. So that's where you can appreciate the funiculi and the fasciculi, no, as they go up or down along the spinal cord. Okay, now I introduced the term tract. No? Tracts are specific nerve bundles or fibers in the white matter of the central nervous system. So their central nervous system, they are not peripheral. So these are bundles of nerve fibers with a common origin and termination within the central nervous system. So meaning common like designation of source and ending. So as a location. So by convention, the tracks are named may be named according to that common origin and as the first stem and the termination as the second stem part of the. So for example here, when you say lateral corticospinal tract, so the corticospinal is made up of two word stems. No? So the first word stem is cortico. It refers to the cerebral cortex. So that means that's the origin. So that's where the tract starts. So that's usually where the nerve cell body is. So it's the nerve cell body is in the cerebral cortex. And the second part of the term is spinal. So that's the termination. It ends in the spinal cord. So corticospinal tract refers to a tract that originates from the cerebral cortex and terminates in the spinal cord. And the lateral one describes in its location around its core. So now these stems are not interchangeable. So you cannot say lateral spinocortical tract. That's a different thing. So although there's no such term, but let's say there's such a term as the spinocerebellar tract. Uh, and there's also another one like cerebellospinal. No? So those are two different tracts. No? So because of that, the convention, the first uh, part of the word refers to the origin, the second part to the termination. Okay. Now, some tracts are described as either ascending or descending with respect to the direction of the nerve impulse. So, if you say ascending, the nerve impulse comes from the periphery up towards the brain. So, therefore, it is ascending. So, if it's going towards the cerebral cortex, it's ascending. Descending would be carrying nerve impulses away from the brain or the cerebral cortex downwards towards the periphery. So the example here is the corticospinal tract. So, so since the impulse here in this image, so it starts in this drawing in the side. So the nerve cell body is in the cerebral cortex. The axon goes down along the through the cerebral peduncle to the brainstem down to the spinal column. Okay, so after crossing the midline here, which is called the decussation, so it goes down to the particular part of the spinal cord to stimulate the neuron in the spinal cord to potentiate or to actuate the muscle, no, motor component. So that is a descending tract. So the lateral corticospinal tract is a descending tract because the impulse is going away from the brain towards the spinal cord to the periphery. Okay, so that's an ascending tract. Now, Gray matter represents, so that's white matter. We'll go to the gray matter. Gray matter refers to nerve cell bodies primarily, and they have no myelin, so they do not appear white. In the brain, there are two areas where they are found. So this look at the upper drawing. So first is the cortex. So that's the periphery. So the peripheral on the surface of the cerebrum in this image. You don't have a cut on the cerebellum, but it's a similar system where there's a cerebellar cortex. When you say cortex, cerebral cortex, cerebellar cortex, that's what it refers to the gray matter on the periphery. Also, in the brain, you have deep nuclei. So the nuclei that are in the dark cuts there in the middle, so deep in the cerebrum. So you have the basal nuclei. We talked about them. The thalamic nuclei here, the thalamus. So and then also in the brainstem in this cut, there are cranial nerve nuclei. No, so that receive the impulses from the sensory cranial nerves and from the impulses from which for the motor cranial nerves. So that's where they originate. So the so there are cranial nerve nuclei as well in the brainstem, gray matter. Now in the spinal cord, as I mentioned, there's a reverse in the arrangement na periphery nga ang white matter, but then the gray matter now is centrally located. It's butterfly shaped, organized into horns. So there's a ventral horn and there are a pair of dorsal horns and a pair of ventral horns. And depending on the cut is, there may be intermediate zones, lateral horns also in the intermediate zone. So we'll talk about that a bit later. So yun, so that's the organization in the of the gray matter. So let's see here. This poll question, don't put the poll anymore. Just answer this. Thalamus is made up primarily of A, myelinated axons, or B, nerve cell bodies. Either chat box or just speak up. Yeah, I mean B. Okay, nerve cell bodies, because as I mentioned just earlier, they are made up of the nuclei in deep within. They are nuclei within the brain and within the gray matter. Thank you very much. Okay, let's go. So now let's go to the central nervous system. Talking about first the brain. So the brain is divided into three major parts: the forebrain, midbrain, and hindbrain. The forebrain includes the cerebrum, what we commonly refer to as brain in layman terms. Okay, it's made up of the outer cerebral cortex, the inner cerebral white matter, and the basal nuclei. No, so inside. Also, uh, the forebrain also contains the diencephalon. So it's not really part of the It's not. Uh, it's still forebrain, but not the cerebrum. So diencephalon is made up of the thalamus, hypothalamus, epithalamus, which is where the pineal body is, or pineal gland, remember in endocrine, yeah, and subthalamus. No? So there's one of the, we could say like one of the minor, or well, a small one, small parts of the diencephalon. So, but the main bulk of the diencephalon really is significant is the thalamus and the hypothalamus here in this picture. Okay, so that's forebrain. The midbrain is just midbrain that connects the forebrain and the hindbrain. That's already part of the brainstem. Now, the hindbrain is the rest of the brainstem, which are the pons and the medulla oblongata, plus the cerebellum here in purple. No? So the cerebellum that just sits on the dorsal aspect of the brainstem, just inferior to the posterior cerebrum also. Okay, so um, now the reason for this subdivision lies in the development of the central nervous system lecture with regard to the embryonic neural tube development, no? That subsequent lecture that will be taught by Dr. Uy, no? So that's so how why the why is this division like this? So this why these parts develop in such a way in this way. So just remember that which part belongs to which at this moment. Let's go talk about the thalamus a bit. So there's that pair of thalami located on either side of what is called the third ventricle. That is a fluid-filled cavity in between the two cerebral hemispheres, right and left. Right, so in the middle, and they are connected to each other by what's called the interthalamic adhesions or connections. So just look at that image there, so in the middle. So they're like some that connection. Now, this each thalamus is primarily composed of nuclei or nerve cell bodies, no, deep in the brain, and they are divided into three nuclear groups or three parts. The anterior in yellow here, medial in blue, and lateral in red. So by a Y-shaped internal medullary lamina. So yun. So that's what that Y-shaped structure that separates these three nuclear groups is the internal medullary lamina. Within the internal medullary lamina, also there is an intralaminar group of nerve cell bodies called the intralaminar nuclei, which has some function as well. Uh, but because it is predominantly made up of nuclei, nerve cell bodies, they appear this the thalamus appears as gray matter. Now the hypothalamus, on the other hand, so just above it. So you see that in this picture you have this oblong thing and the smaller oblong thing there. That's the bigger oblong thing would be the thalamus, and then the small one is the interthalamic adhesion. Below that, that's the hypothalamus. We have these different colors, no? So this region controls and integrates the autonomic nervous system and the endocrine systems and plays a vital role in maintaining body homeostasis. That is controlling our emotional states, fat, carbohydrate, and water metabolism, body temperature, genital function, sleep, and food intake. No? And also the release of pituitary hormones, because that's why it's closely associated with the pituitary gland, as you see here below, no, below the hypothalamus. So they release they have the releasing hormones to the pituitary gland. So that this will you'll have a lecture on the role of the hypothalamus towards the end of the course. Now, let's talk about the cerebellum. Cerebellum stands for little brain in Latin. So it's part of the uh, hindbrain connected to the brainstem at the pons by means of the three cerebellar peduncles, superior, middle, and inferior. And among its main anatomical features, it's divided into three lobes: anterior, posterior, and flocculonodular. Um, and there are two hemispheres, right and left, with a midline vermis. So that little uh, vertical like column part, that's the vermis. It's like the cerebrum. It has folds on the surface, as you see in this cut. So they are like in here instead of gyri, it's called folia. And on the also within there, because of this cut section, you see there's a cortex, and there are also deep nuclei like the basal nuclei in the cerebrum. So these are the deep nuclei of the cerebellum. Okay, you'll learn more about that in another the lecture on the cerebellum. Okay, now this one again, acclamation. The thalamus is part of which division of the brain? Answer: A, forebrain; B, midbrain; C, or hindbrain. Tama. So forebrain. Tama. Remember forebrain. It's part of the diencephalon, which is part of the forebrain. Thank you. Okay, now this one, let's just go to the cerebrum. The cerebrum is divided into six lobes, and four of those lobes are named according to the skull bones associated with them. So just take note there. Remember the skull bone in the head and neck. So there are the frontal bone, parietal bone, temporal bone, occipital bone. So the lobes that are associated with those bones, just aside beside them, those are the lobes. Okay, in the brain. Now, how do but it's okay, you have this brain specimen right there. So, but you don't have a skull bone there. How do you know which is the boundary between these lobes? Okay, so just remember these particular structures. First and most important structure to identify when you have a brain specimen, it's the central sulcus. That's the one in yellow that I wrote there. So that separates the frontal lobe that's in blue from the parietal lobe that's in red. Okay. So that's you see the central sulcus more prominently on the lateral surface of the brain. In the medial surface, it only goes a little bit on the top, and then you just have to go make an imaginary line, project that one down in the medial surface. So the middle view is the one below. The lateral view is the one above in this image. So yeah, so that separates the frontal from the parietal lobe. Now, the other structure, that important structure on the surface of the brain is the lateral fissure of Sylvius or lateral longitudinal fissure or Sylvian fissure, which is this one in magenta. So this one separates the temporal lobe, which is in green, yun down below, no, from the frontal and parietal lobe and the anterior part, no, of the parietal lobe. So the frontal lobe separate talaga, but the anterior part of the parietal lobe is the one that separates it from the temporal lobe. So yeah, that's the lateral fissure. Okay. Now, the boundaries between the posterior parietal and temporal and the occipital are a little bit indistinct, somewhat arbitrary here. But you start off here with a looking at the medial surface. There is a structure called the parieto-occipital sulcus. Yun, that's the solid green one. So you see that mainly in the median, a medial surface. Okay. Now this only goes so far. If you uh, if you project a line towards what is called the pre-occipital notch or temporo-occipital incisure, that small indentation between the temporal lobe and the occipital lobe. So that's the pre-occipital notch. So you make extend that sulcus downward, and then you also make another projection on the lateral surface to connect the two on the lateral surface. So that would separate the occipital lobe, which is in yellow, from the parietal and temporal lobes. Okay. Now, you still have a part of the posterior part of the parietal and the temporal lobe to contend with. So you just make a perpendicular line from the line made by the parieto-occipital sulcus towards the tip of the lateral fissure. Yun. So to separate the parietal lobe from the temporal lobe. So yeah. So those are the borders of these four lobes of the cerebrum that you see in the surface. Okay. So yun. So those are the four. So remember, so I said four lobes are named according to the skull bones, but there are two other lobes of the cerebrum. So what are these? So these are called synthetic lobes because they're not immediately visible on the surface. The first one is the insular lobe. That's the one. So So the insular lobe or central lobe or in island of Ryle or insula may be seen if you separate the lips of the lateral sulcus. So in this picture, yun, there are these two clamps there yun, to separate the lateral view. Then you see it underneath of the lateral of these lobes because they are beside the lips, opercular meaning lip. No? So opercular portions of the parietal, frontal, and temporal. So those are the ones that you separate to see the insular lobe underneath. Now, then what about the limbic lobe? So the limbic lobe is more of a functional system. No? So and in which and the structures that are involved in the system together form that limbic lobe, and which are here in this image below in blue. So even if it looks like anatomically part of the parietal, frontal, temporal, functionally they are part of the limbic lobe. So they are more in the central part of the brain. So these are the structures that come from part of it: the cingulate gyrus, parahippocampal gyrus, the isthmus, hippocampus, dentate gyrus. So um, is later on you'll just identify them when you look at the brain specimen. So it's just these are these areas here. So those are that's the sixth lobe of the cerebrum. Okay. Now on the lab, you will be given a brain specimen to study, and from there you should be able to identify these main features. So you have here in this view, identify the central or sulcus. Let's see here. The central hard thing about this, I cannot do a pointer. Anyway, wait, I will annotate na lang para to make sure you get it. Okay. So this is the yeah, that's the central sulcus. Okay. That's the first thing that you have to identify when you're given a specimen. So the anterior or the frontal part, frontal lobe is this part. That's the frontal. This is parietal. Okay, posterior frontal is anterior, parietal is posterior. So now precentral gyrus is this one. It's the gyrus just before anterior to the post up to anterior to the central sulcus, whereas the postcentral gyrus is here, the one posterior to the central sulcus. There is another groove here between the anterior to the precentral gyrus. That is the precentral sulcus. And then as by terminology goes, this the sulcus just posterior to the postcentral gyrus, this one, that is the postcentral sulcus. Okay. Take note that this here is your lateral, lateral sylvian fissure. Okay. Oops. So again, now on the median surface, you have this big structure called the corpus callosum here. This is the corpus callosum, okay, made up of a rostrum, genu, body, and splenium. Yeah, okay. There is the gyrus above it. This is the cingulate gyrus, and then the cingulate sulcus. This is the parieto-occipital sulcus. There's another groove here that is very important. This is the calcarine fissure, the important feature in the occipital lobe. Okay. Uh, this is the part of the central sulcus. Okay. This is the area of the third ventricle. You have the thalamus. This is the hypothalamus here, and then the thalamus in the area of the third ventricle. Okay. So inferior view, there's another fissure that you haven't seen, but it's there. This is the midline interhemispheric or longitudinal fissure here. Yeah, dividing the right and the left. So you can barely see the corpus callosum there. This is a cut section of the midbrain. Okay. You have the cut olfactory. This is the olfactory bulb, olfactory tract, optic chiasm. Okay. This is the parahippocampal gyrus here. Yeah. With an uncus up there. So yun. So which are part of the limbic lobe. Okay. Then in superior view, you have the again the interhemispheric sulcus, fissure or longitudinal fissure, and here you could also see the central. When in this image, you have to identify really the central sulcus, which is this one. This is the most prominent one. Yeah, that's the central sulcus. So this would be frontal lobe, parietal lobe. Okay. Yeah. So therefore, this is your the pre-central gyrus and the post-central gyrus, anterior posterior to the central sulcus. So you have to sort of like practice this in different specimens and take note that the central sulcus may not always be central because of the way anatomically each specimen is. So, but the important thing identify is which identify the one that actually goes longest and consistently down to form the two gyri, anterior to posterior to it. Okay. Uh, okay. Oops. Ayan. So here again, acclamation na lang. Which structure separates the frontal and parietal lobes of the cerebrum? A. Calcarine fissure, B. Central sulcus, C. Cingulate sulcus, D. Lateral fissure. Ayun, yun, sagot agad. B. Central sulcus. Dapat hindi kayo magkamali dyan. Thank you. Okay. Now, there are different areas in the cerebral cortex associated with specific functions. Sensory, motor, speech, vision, etc. These are called functional cortical areas and they have different terms. May primary, may secondary, may association. So you will learn all that in more detail in the lecture of Dr. Rivera next week on cerebral cortex. So basta functional cortical areas. And there are particular areas of the brain, the cortex, where these particular functions are. Such that, important yan, because if that particular area is affected, you can manifest with these cortical abnormalities or you have problems with motor or sensory or even performing particular functions. Speech or understanding speech, yes, etc. So, yon. So that's the functional cortical area. Related to the functional cortical areas are the Brodmann areas. This is named after this guy, this neuroanatomist, Corbinian Brodmann. He did the histology of the cerebral cortex and saw particular patterns, no, in particular areas of the cortex. And then he realized that these he numbered them according to the type that the uh, in the order that he identified them. So he identified 47 of them. There's a particular number for them. Then he realized that some of these numbers are associated or correspond to different functional cortical areas. Okay. So you will find out that there's certain functional cortical areas. What is this broadband area? There's a particular number. For example, the brain for the precentral gyrus functional cortical area is the primary motor area yon. And Broadman area, that's Broadman area 4. Yeah, so number 4. Yeah. So yun. This green polka dot area, that's in this image. Yeah. So that's the precentral gyrus, and that's the primary motor cortex. So that's the functional area. Number 4, yung Broadman. So you'll learn more about that in the lecture also of Dr. Rivera. Now, another concept we talk about is the homunculus. The homunculus is a schematic representation of the human body, so like a human representation on a specific area of the central nervous system. That is, the human body is projected upon that specific part. Like in this case, the sensory and motor homunculi. So that means in these particular areas, they represent particular parts of the body for sensory and motor. So in this case, the larger the area, the way it's shown, the larger the representation. So from the one on the left is sensory, the one on the right is motor. So um, pre-central yung right, post-central yung left. Gyrus. No, so it shows you how big the face is, so how much sense for the sensory, how much sensation nerves are represented, no, for the particular part of the face, especially the lips and the hands from that image. And then you look at the motor on the right side, the same through. So the way for facial expression, the motor there, as well as the manipulation of your hands. So motor control is higher representation there as compared to the uh, lower extremities, which is just there located in the medial aspect of the cerebral hemisphere. So yun. So that tells you not only the representation but the location of where the nerve cell bodies that are represent these particular areas uh, are or organized. And so if there is a particular deficit in that particular part of the brain, then that will manifest with problems with motor or sensory uh, function for that particular part of the body. Yeah, so that's where the importance of this localization. So okay, ayan. So number 5, ayan. Sagutin nyo nga. Which of the following structures is a schematic representation of the human body on a specific part of the central nervous system? A. Broadband area. B. Functional cortical. C. Homunculus. D. Insula. So C. Ayan. Very good. Ayan. Okay. Ayan. So, let's see. Ayan. Very good. So, we barely have much. 50, we have 15 minutes. Do you have any questions so far na lang? Muna. Before we go to this part of the brainstem. Wala. Okay. Let's go. So, brainstem, uh, first, we'll go first with the external features, and then we go to the internal quickly. We'll have a more better chance of that in the lab. No? So anyway, the brainstem is made up of 3 parts: the midbrain, the pons, and the medulla oblongata. In the lab and practical exam, you should be able to identify the structures on the external surface. And to do that first, uh, you have to distinguish which is the dorsal or ventral surface. No? So yun yung importante. No? So, because you have to orient yourself. What am I looking at? Is it the back or the front, or anterior or posterior part of the brainstem? Now this one is the dorsal view. You're looking at it on the back, no? Because that's where the cerebellum is. So, pero but in the lab, you'll most likely not see the cerebellum anymore. It will be cut off. So what are the main features of the dorsal view of the brainstem for you to know that it's the dorsal view of the brainstem? You have 4 sets of 4 dorsal columns, yun sa baba, yung dorsal columns. That these are the same dorsal columns on the spinal cord. You have they terminate at 4 tubercles. Yan, yung 4 na yun. So they terminate and then knob there at the tip. Okay? And there is a fourth ventricle. which is this diamond shaped area, where it's also referred to as the rhomboid fossa, because it's rhomboid shape, no? So fourth ventricle, four sides, yeah, number four. And then in the area of the midbrain, you have the colliculi, two superior and two inferior. So four colliculi. Yeah. So yun yung so that you know that that is the dorsal view. So what are the other things that you could see on the dorsal view? You see the cerebellar peduncles, yun, the three pairs, the superior, the middle, and the inferior cerebellar peduncles. Yun, they connect to the cerebellum from the brainstem. Okay. Also, on the dorsal surface, you see the origin of cranial nerve 4, which is that one. Yeah. If you are lucky to see it in the specimen, yeah, unless it's cut. So, but you'll see it. That's the only cranial nerve that comes out of the dorsal aspect of the spine of the brainstem. Okay, there are medullary villi to cover the space of the fourth ventricle because it's fluid filled with cerebral spinal fluid. So you have the brainstem and the cerebellum, but there has to be like a roof to cover it. So that's the medullary villi, the superior inferior or anterior posterior medullary villi. Now, on the floor of the rhomboid fossa in the fourth ventricle, you have the medial eminence, this like a column thing in the middle on either side, and the facial colliculus, which is this fat termination there underneath, then below, no, towards the inferior part. On its side, to the border is the sulcus limitans. Okay, that's the important line there, groove there on the side of the medial eminence and the facial colliculus. Its importance in the development of the cranial nerves and is will be discussed later and also in the lecture in the development of the central nervous system. There is also called the structure called the striae medullaris. They divide the rhomboid fossa into cranial and caudal. So they go laterally towards the lateral recesses, where you have the foramina of Lucha, where cerebrospinal fluid exit the fourth ventricle to go to the subarachnoid space. There is a vestibular area. That's the area lateral to the medial eminence and the facial colliculus. It's called vestibular area because the underneath that are the vestibular nuclei for the vestibule for the balance, no. And then below the striae medullaris are the hypoglossal and the vagal trigones. Okay, so underneath are the hypoglossal nuclei and the dorsal vagus nucleus. Okay. The the tip of the rhomboid fossa is obex, no, where there is a hole where you have the foramen of Magendie is where the fluid exits as well, cerebrospinal fluid exits to the cerebello, to the cisterna magna. Yeah. So those are some features of the dorsal view. What about the ventral view? So this is different naman. So from the dorsal view. So what are the landmarks? There are four major landmarks again here in the ventral view. You have the pyramids and the olives. So for pyramids, there is the last arrow that I put there. That's where there's a decussation. There are fibers crossing the midline on either side of the pyramids. If the cut of the brainstem is low enough, then you may be able to see it. Okay, then you have the olives on the either side. So parang there's a potentially magkakamali kayo with regard to the columns of the dorsal columns in this, but this is more short and also you have to look at the other features, no, to rule it out. So the big one here is the base of the pons. No? So there are no other features. It's just smooth Yeah. And then the cerebral peduncles instead of the uh, colliculi are there such as. Plus the exits or the origins of the different cranial nerves. So remember the 12 cranial nerves are from olfactory to hypoglossal, but in the brainstem only the third down to the 12. No? So wala yung first and second. Okay? So you'll learn that more better in the lab. Okay. The question, which of the following structures is best found on the dorsal surface of the brainstem? Dorsal ha, dorsal. Base of the pons, cerebral peduncle, pyramid, or superior cerebellar peduncle. Yan. So, kuha nyo D, superior cerebellar peduncle. Kasi yung base of the pons, cerebral peduncle, and pyramid, ventral surface yan. Very good. Okay. This one I will skip na, different cross sections. Better to learn that more in the video and in the lab. Just remember there is the main regions on the brainstem. There is a tectum, tegmentum, and base. The only one that with the tectum is the midbrain. Okay? Kasi yun sa pons and medulla oblongata, nandun na yung fourth ventricle. Wala, the cerebellum na yung taas nun. So there's no roof. So the only one with the roof is the midbrain. Yeah. So midbrain looks like a pair of shorts. You just take note of the rostral. So main features, I think I mentioned this in the lecture, in the video. And then middle pawns, Princess Leia in the New Hope, yung parang may ensaymada sa tabi. So that's the one. So uh, that's the middle cerebellar peduncle. Caudal pons is like a ship, also middle cerebellar peduncle on the side. There are different features there. Medulla is like a butterfly, but then different parts is like a depending on the cut. So you have the middle with there's no more fourth ventricle in the middle. And the caudal one, you have the decussation of the pyramids. Yun. So these features, read on them and also look at them in the video in more detail. So question here, which of the following brainstem segments has a tectum? So sabi ko na, midbrain lang ang may tectum. So A. Yan, walang pons, wala sa medalla. Okay, very good. Now, this is the neural tube development thing. So that's where the sulcus limitans comes in. Divides the neural tube into an alar plate and a basal plate. Because of the development in the spinal cord, people go laterally, no? Parang ganon. So, sa spinal cord, hindi gumagalaw. Pagdating sa brainstem, they go move laterally like this. Yeah. So that's such that the alar plate parts go lateral and the basal plate part is more medial. The more medial they are motor. The the alar plate is sensory. So naging lateral. So remember, pagdating sa ano naman, sa spinal cord, the sensory part is the dorsal horn, motor part is the ventral horn. So ganun, hindi masyado gumalaw sa alar and basal. So yeah, but then this one in the brainstem, yan, kikita mo, the motor parts are more ventral and medial. Sensory parts are dorsal and lateral. And the sulcus limitans is what separates these nuclei. So that's why in this composite image, which I think is in the video as well, so tells you the location of the articular nuclei in brainstem, cranial nerve nuclei. So the more motor they are medial. The sense most among them, the most medial would be the GSE, general somatic efferents, so which are uh, cranial nerve 3, 4, 6, and 12. And then the most lateral GSA, somatic afferents. Exception is that the pontomedullary junction, the cochlear, would be the most lateral. But on the rest of the cuts, the GSA ones, which are the spinal nucleus of cranial nerve 5, yun, that's the one that is in located most laterally. Now, their authors would want to put cranial nerve 11 thus into general a special visceral efferent instead of general somatic efferent. Yeah, so parang hindi siya, even though it's voluntary motor, it's part of the hindi siya general somatic efferents. Now, you learn more about that functional components in also in the lecture of Dr. Uy. So this is another composite picture where the location of these nuclei are. These are the brainstem rule of 4. I advise you to remember this and to apply. So there are in the brainstem, it's important for regarding brainstem injury, trauma, or infection, whatever. So, and so to like diseases and what is affected. So the four medial structures start with the letter M. Medial, M. Motor, pathway, the corticospinal tract, medial meniscus, medial longitudinal and motor nuclei. So the motor nuclei are medial. Side or lateral are the sensory ones. So sensory, sympathetic, spinothalamic, spinocerebellar. Now, there are four cranial nerves above, at, and below the pons. 1 to 4 above the pons, 5 to 8 at the pons, and this is the nuclear nerves, uh, and then 9 to 12 at the medulla. Now, here, the four motor nuclei in the medial brainstem, most medial ones, can be divisible from 12, so, except for 1 and 2. So, the 3, 4, 6, and 12. So, yun yung pinaka-medial of all the nuclei. So just remember that. So this picture shows you what is the M. So the medial nucleus, the motor nucleus of cranial nerve 12, MLF, medial meniscus, and the pyramid motor pathway, the pyramidal tract. Sensory, that's the lateral S, yan, ng sa tabi. Okay. Which of the following cranial nerve nuclei most medially located in the brainstem? So, so in the brainstem, so 3, 4, 6, and 12. So, alin dyan? So, letter B. Tama. Letter B. So, yun yung ano. Now, oops, time na. May HLC na kayo. Okay. So, just remember, it's in the video, spinal cord. Remember the structures. Let's see here. I'll just go to the one particular part that you have to remember. So the developmental changes, you'll get that again in the lecture of development. So at first, magka-equal yung spinal cord with the column, but then the spinal column grows faster. So that means at birth, the tip of the spinal cord is at level L2, L3. As an adult, it's at level L1, L2 already. So there's a space here under applicable for lumbar punctures to avoid hitting the spinal cord when you insert a needle there. Also, so there's also this figure, it's in Snell, vertebral versus spinal cord segment levels. Okay, so this is on the other side. So how do you know what corresponds to each vertebra? No? So anong spinal cord segment yung tumatapat doon? So yun. So this, just remember this table. Okay. So at the term infant, it's L2, L3. Okay. Now this one, you learn this in the lab. You have to know whether it's cervical, thoracic, lumbar, and sacral, and by different features. So I also mentioned it in the lecture. So the cervical and lumbar have big ventral horns. And you only know the difference with the two because of the amount of white matter. So the cervical has the greatest amount of white matter. The least amount is in the sacral. And then you have yeah, the width of the, take note, the size of the proportion wise, okay, there is a greater proportion of white matter to gray matter in the thoracic kasi maliit lang talaga yung gray matter mo as opposed to the cervical. And then you have lateral horns in the thoracic. Although upper lumbar, you may have it, but uh, because of the ventral horn, it encroaches it. You don't see the lateral horn in the lumbar one. So in principle, if you see a lateral horn in the lab, we will just say that that is a thoracic. Okay, I won't ask this anymore. Just read on the third laminae in the lecture, in the video. Okay, wala na. Any question? Ah, just a moment. Just remember, at the end of the course, you'll have a quiz on regarding neuroanatomical localization. You will be given a case, and you should identify the symptoms, the pathways, and then the most likely location. Assuming that there's only one lesion. That's at least that's as the most that we can ask of you. Okay, thank you for your attention. Mahuli na kayo. Any questions? Okay, thank you very much for your attention. If you have anything that you think I can think